In the brain of a person with Alzheimer's disease, you'll see clusters of toxic amyloid beta plaques building up between nerve cells or neurons. And inside neurons, tau proteins clump together, causing cell death. Ideally, we can use drugs to remove this toxic buildup, in turn reversing the cognitive damage. But this drug delivery is hampered by the blood-brain barrier, which acts as a strong shield, stopping drugs from penetrating to where they're needed. The work in Professor Gotz's lab involves using ultrasound to deliver a treatment through the blood-brain barrier and has been shown to work in mice. This is how it could work in humans. First, the patient is injected with microbubbles. The combination of these microbubbles and ultrasound exerts mechanical forces on blood vessels and temporarily opens the blood-brain barrier. This then allows drugs and antibodies to reach amyloid plaques and tau tangles and ultimately clear them. What's more, the research at the Queensland Brain Institute has shown that ultrasound alone clears the plaque and tangles before drugs are even administered. This work has enormous potential for reversing one of the most pressing health issues in a rapidly ageing society.
Translational research involves starting with a good idea in the laboratory, something that might have been discovered or developed in the test tube or the petri dish, and perhaps tested in small animals, mostly mice, and perhaps some larger animals as time went on, and then finally getting it into the clinic and testing it in humans. Medical interventions for any disease can't be given to humans by the local doctor unless they're approved by the regulatory authorities, and they won't be approved by the regulatory authorities unless all the steps in the development pass muster. Now, this process is a very detailed and expensive process. It takes about 15 years from the good idea being discovered in the laboratory to it being effectively tested in the clinic. And the testing in the clinic goes through a number of stages. The first stage, usually called phase 1, is when we test a new treatment for its safety. Can it be safely given to humans? Lots of things can be safely given to rodents, but rodents are tough little critters, and humans can't tolerate the same things that rodents can tolerate. So we have to establish whether something is safe and tolerable in humans. Secondly, in phase 2, we have to establish that it works in humans, at least in some humans. And then in phase 3, when we do larger clinical trials, we have to establish just what the limits of the treatment are, be clear about what the dose is, how long the treatment should be given, and monitor large groups of people for rare side effects. So it's quite a complex matter. We've already heard, I think, that to develop the ultrasound technique, which QBI is involved in developing at the moment, over the next 5 years alone is going to take probably $30 million. So it's a huge investment. The ultrasound system has been tested in a mouse model of dementia and found to be effective. It's also now been tested in a sheep model, and the sheep tolerated it quite well. And so the next step starting next year, 2019, would be to extend this work into humans. Initially in a small number of humans, possibly as few as 10, in order to establish the feasibility and the tolerability and the safety of the technique. If it proves to be tolerable and safe, then we can move to a much larger clinical trial where larger groups of humans are given the ultrasound technique. And as Rebecca's said, the ultrasound technique can open up the blood-brain barrier briefly to allow bad things to come out, but also to allow good things like antibodies and other treatments to go in. And so that's the purpose of that technique. And because the blood-brain barrier is only opened up for a short time, we're hoping that not too many bad things are going to get in, that only the good things are going to get in. When I say bad things, I'm thinking about bacteria and things like that we don't want to get in. And that's one of the main reasons we have the blood-brain barrier. So a lot of work is required, and next year we hope to start recruitment probably in one of the major hospitals in Brisbane of people with relatively mild dementia of the Alzheimer's type to see whether the ultrasound technique works as well in humans as it seems to work in mice and sheep. So in phase 1 we're looking primarily at tolerability and safety, not at efficacy or whether it works. In phase 2 we'll be primarily looking at efficacy, does it work? And so then the question is, well, how do you measure whether it's working? I don't think anyone's going to be interested in the treatment unless there's clinically obvious improvement, you know. I don't think people are going to be satisfied with improvements that you see on a brain scan but that don't produce any improvement that the patient or the family can see themselves. So we have to use measures of the actual clinical improvement of the patient. These are bound to be things that we apply to the patient themselves, tests and interviews, and also questions that we ask the family about their observations about how the patient's going. So we're going to have to use clinical material plus Gail's neuropsychological testing to establish whether there's been objective as well as subjective improvement in the patient. We'll also be doing sophisticated brain scans to see if the proposed mechanism of the improvement, if it should be found, is as we think. You know, that opening up the blood-brain barrier, delivering antibodies or whatever modality of intervention we are delivering through the blood-brain barrier is actually the reason that the patient's experiencing improvement. So to do that, we have to do randomised controlled trials against placebo. But to start with in phase 1, we don't need to do placebo because we're not really looking at efficacy, we're looking at safety and tolerability, so it's a different kettle of fish. So it's quite a long road, this road of developing a new sort of groundbreaking technique to deliver therapeutics to the brain. We have to do it in a very careful and measured way because people are very attached to their brains, aren't they?
Thank you. So I think now at this point we'll open up to all of you if you have any questions about any aspect, whether it's the ultrasound project itself or other aspects of dementia and Alzheimer's research here at QBI. Is there anyone who would like to start off?
My understanding is with the ultrasound opening up the blood-brain barrier that it has potentially many clinical applications, not just for Alzheimer's but all sorts of other neurological issues. So perhaps if somebody could answer that.
That's great. Thanks very much, David. Shall I start with Rebecca or Gerard? Who would like to take that one? Gerard would like to take that one.
Thank you. Yes, that's right. So if demonstrated to be safe, it would be a generic technique which could be applied to a number of different neurological disorders based on the same principles of removing things which shouldn't be there or targeting, better targeting drugs or antibodies to the site that they're needed in the brain. The ultrasound devices can be linked, they can be phase linked, and they can be tuned to fire a beam at a certain location in the brain, a certain depth in the brain, so can target different brain disorders which have a predilection for different parts of the brain. Particularly, many disorders which ultimately cause dementia affect the whole brain in the end, or pretty much the whole brain, but they often start in a single or a couple of relatively localised areas in the brain. So for argument, you could target those areas early in the course of disease in an attempt to stop the disease spreading. So yeah, I think the technique has potential general application. It makes sense, though, to start with a very well characterised disease like Alzheimer's disease to prove that what we've seen in the other animals also works in humans. But you can imagine frontotemporal dementia, motor neurone disease and the like might be additional targets for this sort of approach. Potentially Parkinson's disease as well, and its associated conditions, Lewy body disease perhaps, but so yeah. I think the value of the technique is that it's a technique with potential wide applicability.
I was going to add to that, yeah, a technique with wide applicability and the ability to apply different therapeutic agents targeting different aspects. So there may, whilst Rebecca's work is targeting tau and amyloid beta, perhaps there would be other targets, other antibodies, other agents to be perhaps drugs that don't by themselves manage to cross the blood-brain barrier, but could improve their efficacy by being delivered along with ultrasound.
Hi. It's just follow up to that question, more a practical curiosity question. You mentioned, I think it was Jared, that you have a short window in which to target, to go through the blood-brain barrier into the brain. You said for a short while, I think you both said. So does that mean, how short, what are we talking about here, 5 seconds or 5 hours? And if you're looking at getting a cohort of people next year, how are you going to design that kind of intervention? Is it something that you do to somebody every day or over a period of time, or how do you know that it's working? Sorry.
It's a good question. We expect the blood-brain barrier to open up for minutes to hours, not days to weeks, and we expect to have to apply a series of treatments. Now, the precise interval between the treatments and the ultimate number of the treatments yet to be determined. But it's bound to be a course of treatments over an extended period of time, separated by a gap so that we can observe the patient for their response or any toxic effects. In phase 1, where we're looking at the safety and tolerability of the intervention, it'll be a very experimental approach, yes, yeah. And it needs to be, because we need to keep an open mind about things like dose and frequency, etc.
I can just follow up on that also, just in terms with antibodies. Antibodies have about a half-life of about a week or two. So, for example, once you get them into the brain, they should be functioning in there for around that for about a week. So you do a weekly treatment would be what you'd probably start with antibodies. Thank you.
I just wonder, with the 10 to 15 year delay and a definitive diagnosis, is there any opportunity either with blood test for abnormal proteins or cognitive tests to pick it up much earlier? Are there any screening tests becoming available?
I think in terms of the cognitive tests, those are getting developed all the time. But I would just clarify, the delay in diagnosis from symptom presentation isn't the 15 years. That's sort of more like 3 years at the moment. But, I think that part of it is education as well, and having these things out there with general practitioners, not just specialists like Jared, who knows exactly what to look for. So he's picking it up earlier in that 10 to 15 year phase, but other specialists aren't. So there's a few prongs we can work on here.
The other thing to add is that even before someone develops any symptoms at all, you can, using tools that are mostly used in research rather than in normal clinical practice, establish whether someone has more amyloid in their brain than they should have. And there are 2 ways of doing that. One way is to do a amyloid PET scan, PET scan. We do PET scans for cancer all the time, but they're not amyloid PET scans. They're a different sort of PET scan. So you can do a specific sort of PET scan that looks at how much amyloid is in the brain of a human, and you can establish a threshold, and if the amyloid is above that threshold, you can say the person has too much amyloid and they're at significantly higher risk of ultimately developing Alzheimer's disease. Will they develop it? The test doesn't say with 100% certainty that the person will develop it. It just says they belong to a group at much higher risk of developing it. So it's a probabilistic thing. Secondly, you can put a needle into the spinal canal, draw out some cerebrospinal fluid and send it to a reference laboratory in Melbourne, and you can measure the ratio between the 2 proteins, amyloid and tau, and that can tell you whether someone has abnormal levels and almost certainly has Alzheimer's disease. Now, the confusing thing about all this is that we're used to thinking of Alzheimer's disease as being dementia, but as Gail said, you can have Alzheimer's disease and probably all the other neurodegenerative disorders for decades before you have any signs, any clinical signs. So I think we have to be very careful about not conflating the term Alzheimer's with the term dementia, because dementia is only the very final phase of a very long, drawn-out process that goes on in the brain. The brain's got an extraordinary amount of reserve capacity, so it can tolerate a certain amount of damage. It can tolerate strokes, it can tolerate a lot of damage from Alzheimer's disease before there's any sign of dementia. So the ultimate aim is to treat people who've been demonstrated to have Alzheimer's disease but don't yet have any sign of dementia with effective treatments. That would be the aim. For the last 25 years, we've been testing drugs and antibodies in people with advanced or moderately advanced dementia. They don't work. They all work in mice. They don't work in humans with moderately advanced dementia. So it's back to the drawing board. So really, I think we should be treating people before they get their dementia, not after they get their dementia. And because there's such a long lead time, it should be entirely feasible if we can identify who those individuals are. So I'm sort of more optimistic now than I used to be about prospects. But we do have to have effective treatments, and that's what this ultrasound system is attempting to do.
I think just to add to that, it is the combination. So if we can pick people up earlier now with the methods, with cognition, then you do start to get people at the earlier disease phase. And, you know, there are subtle signs. It doesn't have to be fully blown dementia. So that's what we're trying to figure out is who are the people with even subtle signs and at what point can we identify those?
This brings us to the point, I guess, is when does dementia become dementia? I'd be really interested in a good definition, because as an outsider, I, for example, observe myself, and I would like to know when I was showing signs of dementia as against what my brother calls Alzheimer's disease. And so, but the first half of the question is very serious.
From my point of view, I would diagnose dementia if a person had three things. First of all, they or their family had reported a noticeable decline in their cognitive function over time. Secondly, if we tested them on some cognitive tests of memory and thinking, they performed extremely poorly against what we expect them to perform or what they would have performed earlier in life. And thirdly, that those two things had led to functional impairment in their lives. So if you have some cognitive impairment as you get older, but it has no functional significance in your life, you're still getting on with your life, you've adapted to any small changes you've had in your cognition, you know, you're taking a few more notes and you're keeping your passwords somewhere safe. So if you notice some change in time in your cognition, as we all do, and you adapt to that, that's not dementia. It's the inability to adapt to the change, which means where you fail to adapt. The wheels come off, and then you've got to have external support. That's when you tip over from, say, mild cognitive impairment into dementia, I think.
I agree with everything he said. No, I just wanted to say one of the other things that maybe a lot of you are sitting there wondering, and I get asked by my dad especially all the time, what can I be doing to maintain and improve my function before anything happens? And so that's obviously when we talk about what areas of cognition, what tests. Another aspect is training and enhancing abilities, and I know there's lots of computerized programs and things out there. There's not a lot of evidence that they're about what they actually do specifically, but one benefit is they do focus your attention. So, you can't go wrong with that. If you feel inclined to do it and then you enjoy it, why not? But there's a whole avenue here of research in terms of training to enhance function, particularly about adapting So I know that I have colleagues that are looking into that. So that's obviously for the healthy ageing. It's a preventative, more public health issue. So that is not really about treatments of dementia, but that's really how can we keep our population, our society healthy and adaptive and problem solving, creative, all those things.